Okay, so this is about the last AP free response question we're going to be able to go through before you take the test. Some of you, again, have already taken the test. So when we look at this first question, and some of us looked at it in class already, we see that there's a table. Again, when I look at a table, I'm already thinking, what is the control group? What's the experimental group? What's the independent, dependent variable? What are they comparing? What's the situation? And the table often summarizes, in fact, that's the whole point of a table, is to summarize the results. So it tells you a lot about what's going on with the experiment. So here's his average probability of revisiting nectar source. So without even reading the question, I'm thinking nectar source is some kind of pollinator or some kind of organism that feeds on plant nectar, so flowers. And its probability, so it's going to be either in decimal or percentages. And since this says plus or minus SEX, so when you're using SEX, what you're looking at is standard error. So this is really the standard error. Plus or minus 2 is the standard error for this many samples. XO with a line over it means how many samples you have. And so when you're looking at average probabilities, you're looking at average probabilities from a sample. So obviously you can't look at all the whatever the pollinators are that are collecting nectar, but you're gonna sample. You're going to study a certain number of these pollinators, in this case bees, right? And so you're going to look at how many bees. Let's say you study 100 bees or 1,000 bees. That's not all the bees. So there's going to be some error, right, based on your sample size. So the larger the sample size, the less your error. So a plus or minus 2 of standard error of a plus or minus 2 means that whatever the value is, whatever that percent probability is, you can go plus or minus that average probability is going to be plus or minus the standard error of 2, plus or minus 2. So you have 10 minutes, and here you have a control group. Here you have a caffeine group, or your experimental group. So already I know I have my control group, my experimental group. Here I have a 0.09, so I have 0.72, and I can go as high as 0.81 or as low as 0.63 because of these two values. At this point, in reading the problem, I don't have the details as to what information I need or don't need, but I'm just analyzing the table at this point. I'm thinking I have a different error range for each measurement. Here, this is a 24-hour period. Here's a 10-minute period. So I have to consider, I'm probably going to have to compare these two. And I'm probably going to use a bar graph at this point. I sent you a summary of when to use what graph and how do you determine that. If you look at your workbook, your science math workbook, it goes into more detail how to do that. Hopefully you've been working on that, although this year's been so crazy I can't really blame you if you haven't. So here's a flowering. If you read the problem, it talks about attracting pollinators such as bees and visual cues. Now I highlighted this before I read the questions, and someone in class said, "Why don't you? Is it a good idea to read the questions before we do the reading, before we do our highlighting?" And the answer is yes. It is a good idea to go ahead and read the questions first. I like to read first, but it's probably a lot more efficient to read the questions first and come back and highlight, and therefore read with purpose, right? So that's the idea, is read with purpose. And what could give you the purpose? Maybe look at the questions that the questions you're being asked. So just read the questions first is a good idea. Then go back and highlight the things that you might think are important. All right, so this caffeine is at 0.1 millimolar. That's 1,000th of a molar. A mole is equal to moles per liter. This is chemistry, right? It's a measurement of concentration. Remember that a mole is equal to 6.02 times 10 to the 23rd of something. It could be anything. Number of atoms, number of molecules, what number of shoes. But it's a large, very large number. And so millimolar is 1,000th of that, right? And 0.1 millimolar is even 1/10 of 1,000th of a mole, right, of caffeine. So a very small amount of caffeine. Now, how much is too small? How much is too much? Well, it all depends on the substance, right? So at what point does caffeine become toxic, and at what point is it beneficial? So as with all medicines, as with all chemicals, it's about concentration. So the concentration of a substance often determines how much, harm versus benefit. If this were another class, we would be talking about LD50s. And LD50 is a measurement of a concentration at which 50% of the population die. So you can calculate the LD50 for any chemical. Sugar, for instance, could kill you at a high enough concentration. Theoretically, water could kill you. It would be difficult, but water could kill you at a high enough concentration. So there is such a thing as this calculation of how do we determine, harm from benefit? How do we determine what percentage, at what point does a particular chemical become deadly or just make you sick? And so you do that with all chemicals. You can do that with any chemical. And in this case, we're looking at caffeine versus not using any caffeine. So, but there is a key here, and this is just for a personal point here, and that's that every chemical has a concentration at which it becomes deadly. There is no completely benign chemical. There's nothing that you can take at any concentration and consider yourself safe to do so. Right now, some things it takes a lot to do to create a real problem. Your body has evolved, for instance, to deal with sugar, with food in general, with things like proteins and fats. Your body's dealt with it. Obviously, long term, chronically, would be an issue, but high concentrations in any one meal probably wouldn't kill you if you're healthy. That goes for salt as well. We've evolved to deal with salt. However, if you think about it, a snail can take a lot less. It's evolved not to be able to deal with a lot of salt. So the LD50 for salt for a snail is a lot different from an LD50 for a human. So these are numbers. When you see these numbers, I hope this helps you put these numbers into some kind of context. It's not necessary to understand all this for this problem, but I don't know what kind of question they're going to ask you on your particular AP exam, so it's good just to give you a little background here. Again, this would be more for AP Environmental Science, but it's still a good idea to to have some concept of what it means for a drug to be deadly. So the problem says to construct on the axes, construct appropriately labeled graph and illustrate the effects of the caffeine. I've already determined that I want to use a bar graph, so I graphed it. I graphed it as a bar graph. I graphed the control versus the caffeine, so the control group versus the treatment group, right? The treatment, either it's a control group or it's a treatment group. And here you have probability plus or minus SE, standard error, depending on the sample size. Absolute value over X is a sample size. And then we determined how many lines. There's 20 lines. The maximum number from the previous chart, right, from the chart is the maximum number is, I think, 0.9. So we went all the way to 1. Divided 1 by 20, and we got every 2 lines is 0.1. We put those numbers on there. Then we graphed the bars. I made the 10-minute circles and the 24-hour slashes. I graphed the error bars here, the range of possible heights. So it can be as high as this or as high as that. And this is where it is, right? That's based on the table that from the previous page. And of course, we gave it a title that makes sense. There are no real units in probability. These are percentages in decimal form, so it's 0.1. You could always say, you could always turn this into percentages and then graph percentages if you like. But it's all right just to graph it this way. If you are given data, I don't know if they're going to ask you to graph. It's unlikely they're going to ask you to graph. But if they do ask you to graph and determine the axes, please try to remember to always put units on everything that you actually graph. So now let's go ahead and take a look at this. This is a problem. Again, I'm going to look right away at the graph first. I'm going to see time in hours. I see seed germination in percent. So I know I'm going to be planting some kind of seed. How many of these seeds actually germinate over time? And I'm going to have at least 1, 2, 3, 4 different groups. I can see from the graph without reading any more that I have my control group. So I know what my control group. And now I read the figure, the label, and I know it says the effect of carrageenans, CAR, and trimethyl butylides, TMB, on seed germination in lactuca plants. The error bars represent plus or minus 2 standard errors, right? Okay, so this is plus or minus 2, up or down. So the error is how many seeds germinate in that particular time. You'll notice that the error bars are vertical, not horizontal. So that just tells you that this is about how much this is going to be, what is this? This is going to be something like, for instance, this one right here is probably going to be something like 13 or 14 seeds, 14% germination, but it could be as high as 18% or as low as 10%. Low as 10% germination at 36 hours. So that's what the error bar means, right? That's what these little whiskers mean. They're error bars. So obviously this is trimethyl butylides, and these are the carrageenans, right? So carrageen and trimethyl butylamide. I see that this slope with just the carrageenans, the control group, which has probably neither. I haven't read the experiment yet, but probably neither. You see that it's pretty much they start to germinate after what looks to be 15, 16, maybe even 16 hours, and they have a pretty steady slope, pretty faster than these two, slower than this one. This has the highest slope, so this rate of increase of seed percentage germinating in these in fact almost 100%, the maximum percent germination is reached after only 16 hours. Before In fact, the max is almost reached for the Caricans alone before the control group even starts to germinate. The control group germinates a great percentage before either the other two really get started germinating. I mean, obviously here they're germinating here, but at a low percent, below 10%. Before they even get to 10%, these guys have already reached 25% germination. By the time these guys are at 70%, which is their max, it looks like that's going to be their max. If I kind of extended this line, it's probably going to plateau here or somewhere near 70. Well, it looks like near 75. Then you can see that these two, this one maxed out at about 22 and this one maxed out about 12 or 13. So the control group is doing a lot better than either one of these two. Whatever advantage Caricans give them, it looks like TMB takes some of that advantage away. In fact, hurts them substantially. TMB by itself hurts it the most. So it looks like trimethyl butylides stop or slow slow the germination percentage, harm germination, the seeds, so that most of them don't germinate. Where Caricans help somewhat, the control group can get up as high as 75%. With the Caricans, not only do they germinate faster, they reach max faster, their maximum is actually higher. It's about 92, 93% versus something around 70%, 75%. So I already know a lot without even knowing what Caricans are, the background of any of the biology, but just that graph alone told me a lot. So it says fires frequently occur in some ecosystems that can destroy all the above ground vegetation. Many species of plants in these ecosystems respond to compounds in smoke that regulate seed germination after a major fire. Caricans, KR, and trimethylbutymides are water-soluble compounds found in smoke that are deposited in the soil as a result of a fire. Caricans TMB bind to the receptor proteins in the seed. And as a study, now here's what I'm thinking when I read that. I think of these seeds, right? I think of some kind of seed. And I'm thinking that this seed has some cells in it. If I kind of zoomed in on that seed, I'm thinking that there's some kind of receptor that all right. I'm thinking that there's some kind of receptor here that's going to bind to either the Caricans, KAR, and I'm not sure, let's see, it's bind to receptor proteins in the seed. So the Caricans bind to one receptor. I'm going to go ahead at this point, without reading any further, I would assume that there's some kind of, that the TMBs have another shape, and have their own receptor. And the reason I'm thinking that they're not competing, maybe they compete with each other. Maybe TMBs competitively inhibit CARs. I'm not sure yet, but this is what I'm thinking as I'm reading it. In the study on the effects of smoke on seeds, researchers recorded timing and percent of seed germination in the presence of various combinations of CAR and TMB. The results are shown in Figure 1, and we just looked at that. In the second investigation, the effects of available water on the seed generation after fire, researchers that treated the seeds with KR or TMB. The treated seeds were then divided into two groups. One group received water rinse, the other received no water rinse. The seeds were then incubated along with the group of control seeds that were not treated. The results are shown on the table. All right. So as I go ahead and read this, I'm looking at germination results. I'm seeing treatment group 1 is the control, so there's no TMB or CAR or water, and I have some control result. 2 is a treatment group, has CAR, no TMB, no water, is it different from control group, right? 3, you have, and I'm assuming they're putting different and control result. I'm assuming that what they're same as control and different from control. What I'm thinking is that they're going to have us try to figure out, based on these and this table, how these two interact, these two molecules interact. So this is the control group. This 2 and 3 are different from control. If I add CAR here, this is different from control. If I add TMB, it's different from control once I rinse it. This is a control group where I'm adding water, but not the TMB or the CAR, and it's different from control. When I add CAR, it's different from control, and when I add TMB, it's the same as control. Now, isn't that interesting? When I rinse with water and add TMB, the effects of TMB go away. When I add water and add KAR is still, you're still getting a result. You're still getting something that's different from the control group. So here you see the control group adding TMB is different from the control. KAR and TMB is different from the control, and KR is different from the control. But when I add here TMB, no CAR, and I rinse with water, I get same as control. Now that's interesting. I'm not sure what that all means yet. I have to look and read some more, but it's interesting. Let's do here. It says researchers made the following claims about the effect of KR and the effect of TMB on seed germination relative to the control group. It says KR alone affects the timing of seed germination. Well, I think that's fair. KR alone does affect it. In fact, affects it positively. KR alone affects the percentage of seed germination. So not only does KR increase the speed, like we initially analyzed the graph, we saw that KR actually, they started to germinate quicker. They also increased the percentage of germinated. That's correct. We identified that early on when we first looked at the graph. TMB alone affects the timing of seed germination. Well, see, it slows it down, you see. This is when the control would start. You add some TMB, and all of a sudden it takes a few, maybe a couple more hours for the seeds to start to germinate. TMB alone affects the percentage of seed germination. It does. It negatively affects it. The control is here, and TMB alone does this. Now you see the error bars. It could be, there is some error, some standard error, but that standard error, the results are well within the standard error, so they're fine. I don't have a problem with saying that TMB does negatively impact the percentage of seed germination, and that it slows down that germination, the beginning of that germination, where KR speeds up that germination, and it increases the percent they germinate, that germinate. Provide support for the data from Figure 1 to for each of the researchers' claims. Well, again, what I would say is that KR starts, according to the graph, the KR starts to germinate sooner at 12 hours rather than the control group that starts to germinate at what looks to be, let's see, 1, 2, 3, 4. So 4 is every 3 hours. So it looks like the control group started at 18 hours. They started germinating 18 hours, and the KR group started at 12 hours. That's what I would explain here. And I would explain that difference for each one of those. Basically, what I've already said, I would have written it down carefully, making sure that I would always check that what I wrote, whatever that is, matches the graph. Right? I don't want to write something, I don't want to read the graph, confuse myself, and then write something that's opposite, or maybe write this line's explanation for number 1 and that line's explanation. I got to make sure, I got to double check that if I'm talking about KAR alone, then I'm talking about this one. If I'm talking about TMB alone, I'm looking at this line compared to the control group. I'm always comparing to the control group. There's an intense competition by plants to successfully colonize areas. Why is that cut off? Let's see. Oh, I see. So it says, all right, so it provides, so make a claim about the effect of rinsing on the binding of CAR to the receptor in the seed, and about the effect of rinsing the TMB to the receptor in the seed. Identify the appropriate treatment groups and results from the table that, when compared with the controls, provide support for each claim. Well, it's basically what I already said about this table. So, remember I said this was interesting. I said with these here, I didn't add any water to these. This is what happens when there's no water, no rinse. There's no rinse provided. And when there is no rinse provided, this is the control group. This is what I expect to see. And this is what I see. They're both different from the control group. Now I add water. In these three groups, I add water. And again, here's my control group. In these two that I rinsed with water, I noticed that CAR, the one where I added CAR, is still different from control group. You'll notice that these two are still different. They're still different. And so what that suggests to me is that CAR's not rinsed away. That CAR binds more strongly, or at least the binding of CAR is not easily dissolved. That relationship between CAR and its receptor is not easily washed off. Once they bind fairly strongly, as compared to that between TMB and its receptor. Assuming they have different receptors, even if they're the same receptor, if these are competitive, even if TMB is a competitive inhibitor, the TMB is not binding as easily with water as strongly as CAR does, right? So I can say that CAR. Let me go ahead and write it the way I would write it, and I would reference this graph. So I already told you for A, I would have written that. But for B, I would have written, it says, let me make sure I'm getting this right, about the effect of rinsing receptor, about the effect of rinsing on the binding of TMB in the receptor of the seed, identify appropriate treatment and results in units. All right, so my statement would be, my statement for B would be that rinsing does not change the interaction between, and actually rinsing with water, I suppose, KAR and its receptor treatment group two has a result that is different from the control. Whether without the water rinse. Treatment group treatment group 5 has the same result. Let me rephrase that. Group 5 still is different from the control, even though it is rinsed with water. So I just want to verify that I've answered the question, and I've written it, and I want to make sure I've answered it, and so I can get as many points as possible about the effects of rinsing with binding a car to the receptor. The effect why does it group these things? I don't know. Oh well. So I'm going to go ahead and read. It says, to the receptor seed about the effect of rinsing and binding TMB to the receptor and seed. Identify the appropriate treatment groups and results. A table that are compared. So it said rinsing with water does not change the interaction between CAR and its receptor. Treatment group 2 has the result, a result that is different from the control without the water rinse. Treatment group 5 still has a different result from the control, even though it is rinsed with water. For the non-CAR groups, I would do the same thing, because in order to get the full points here, I would have to describe both the TMB group and the CAR group differences. I'd write the exact same difference, TMB treatment groups, one, and probably 6, but let me double check. 1 and 6, right? You'll notice that 1 and 6, I'm going to compare treatment group, not 1, but 3 and 6, because 3 has TMB and 6 has TMB, right? And I'm sorry, 6 has TMB. And of course, 6 has no water, where group 3, 3 has no water rinse, where 6 has a water rinse. So I would write the whole thing again, but this time, and it's not 1 and 6, but it's groups 3 and 6. And 6. So this is TMB treatment groups 3 and 6, right? And I would say rinsing with water does change the interaction between TMB and its receptor treatment group three has a result that is different from the control. Without water. While treatment group six is rinsed with water. And the result is now the same as the control. So that's, I think that's pretty much all that that's going to be that you need to say. And really all you're doing, if you think about it, all you're really doing is let me just make that smaller even. All you're really doing is explaining this chart. You're reading this chart and putting into words, into sentences, what you see here. You're comparing the rinsing of water and the result of rinsing of water to the car group, the car column, to the TMB column. You have two sets, right? You have the set before without rinsing water, and then after in the group, and the set of experiments where you did rinse it with water. And that would answer your questions and get to your other point for supporting the claim. So see, make a claim. That would get you a point if you made a claim for each. And you get another point if you provide evidence for each. So these two would get you 2 points for B. Is there, the next question in C, it says, there is intense competition by plants to successfully colonize an area that have been recently cleared of fire. Describe one advantage of CAR regulation and one advantage of TMB regulation to plants that live in an ecosystem with regular fires. So this is going to require you to consider how organisms are going to interact and why is anything producing CAR and how does CAR work biologically. Where in the previous parts it was just analyzing, I shouldn't say just, but it was analyzing, in the previous parts you were analyzing the data that was already given to you on a chart. Now they're telling you, well, look, they're saying these CAR proteins, this is CAR is being released by the plant after it's burned, and TMB is released as it's burned by various plants as well. TMB slowed the germination of seeds, where CAR speeds the seeds germination up and increases the number of seeds that germinate. So what kind of interaction, what are the advantages? Why would seeds, and again, look at the question again. There's intense competition by plants to successfully colonize areas that have been recently cleared. So you got to imagine you had a forest, now you don't have a forest, it's all burned down. And you've released CAR and you've released TMB. why would the plants release a chemical that slows down germination rate and another chemical that speeds up germination rate? What's the advantage and what's the disadvantage? All right. So to answer this question, what I like to do is I like to draw these things out. And I've told you that probably, so you probably remember me telling you this in ninth grade. But drawing it out really does help. So I imagine trees. And it's a forest, so there's probably a lot of trees. And then there's a fire that happens, and there's trees, they're competing, they're releasing seeds. They go through cycle after cycle of germination and growth, maturity. There's competition. They're leaving out, they drop their seeds. Let's draw those seeds in pink. They drop their seeds like those little helicopter seeds you see dropping from maple trees or pine cones or whatever. They follow the ground and they get buried. But they tend not to germinate. If they start to germinate now, and once they fall to the ground, remember what one of the number one things that plants need is a sun. Then these seeds that fell to the ground won't be able to grow because there's no sunlight hitting them. And these mature trees are going to, not only are they going to take up all the sunlight, but they have a deep root system. They're going to take up a lot of the water, a lot of the nutrients. So it's really not satisfactory for them to grow. You know, it would be disadvantageous for them to start to germinate now while they have all this competition. So a fire happens. And in places like California where there's forest fires and in Savannah where there's forest fire, these systems have developed to take advantage of this fire that's occurring, that occurs periodically because of dry seasons. By periodically, it used to be, a forest it might happen once every 100 years or so, but in the case of global climate change, that's changing. But in any case, you got this what's left is ash. Now the interesting thing about ash is that it has all this great nutrient, right? This thick, rich nutrient. That's all the leftovers from the burning of all the bushes and trees and grasses. Underneath that ash are still those seeds. Oops. You still have all those seeds sitting there, but no trees. Obviously, you still have the sun shining down. So this is after the fire, right? Time goes by after the fire. Now, when these trees burned, two chemicals were released: CAR and TMB. Together, few of these, while these two are both attached to their receptors. Again, I'm assuming there's two receptors. It could work in some other different mechanism, but whatever the reason, as long as both are present, as long as you have both present, you can go look at the graph, right? And you can see what happens when both are present. You see whether it's just TMB or TMB and CAR, you're going to get really massively decreased germination. Only a few of these seeds are going to germinate. Specifically, when both are present, about 25% or quarter of these seeds, versus 90% when it's just CAR, right? So when these two are both present, only about 20% are going to be germinating, starting to grow little seedlings. and turn into trees if necessary. But what do trees need? They don't just need sunlight. They have the nutrients. That gray, this gray ash is chock full of nutrients. It's right above the soil that was already had enough nutrients to support a whole forest. And the dead animals and the dead bushes, all the burnt up stuff is all really great nitrogenous material, nitrogen, phosphate, potassium is all there in the soil, ready for these trees to grow. But what's the one thing that's missing? What do plants need to grow? We know they need sunlight. We know they need nutrients. They have both of those now after the fire. They have a lot less competition because there's nothing there. So then what else do they need? Hopefully you're saying, and you've said it a bunch of times now, you need water. So water falls, and obviously it's going to rain eventually. If it doesn't rain, it won't, and then nothing's going to grow. But let's assume it rains. Assume that some more time passes by, and you get rain. Along with that nutrient layer and the pink seeds and nutrient layer, and there's some that already started growing. You got rain. Well, what's the rain doing to the car? The car, it stays. The car binds. It's going to stay bound. What's going to happen to the TMB? Well, I hope you're saying that the TMB is going to get rinsed away by the water. And because the TMB is rinsed away by the water, the only thing left at this point is car. And so now you're going to get 90% of these starting to grow because you have the perfect conditions for growth. You have water, you have the sun, you have soil, nutrient-rich soil. So you have everything it needs because there's rain, because there's water, because TMB rinses away after rain. So there's intense competition of plants to successfully colonize an area that had been recently cleared by fire. Describe one advantage of regulation and one advantage of car regulation, one advantage of TMB regulation to plants that live in an ecosystem with regular fires. Well, there's a bunch. You can name, I just described a bunch of them, but let's go ahead and just name one for each. If you said something like, right, so you can, and I'll try to find the answers and email you or post the answers that the AP board said. But until then, we know that car's advantage is by having car present. One advantage of car regulation is that a greater percentage of seedlings grow. I should say germinate. And faster. But germinate. After a fire, reducing competition since there's nothing left. Right? There's no competitors left because everything's been burned away. That's the whole point, right? So that's the advantage. Cars only produce when there's a fire. Well, what's the advantage of having TMB? We said TMB slows things down and it hurts, it inhibits germination. Well, what's the advantage? Well, TMB only allows for maximum germination when there is enough water. So together, these two produce a combination that maximizes the survival of the species, in this case this tree, by making sure that after fire, when there's less competition for the seedlings, they grow quicker, they germinate quicker, more of them germinate, from 20% to something like 90%, and that that maximum growth rate and the maximum growth percentage only happens after a rain and when there's enough water available for that germination, for all these trees to germinate and grow. Again, maximizing the survival, the percent survival of these young trees. All right, so that answered that question. Again, I'll send you the scoring guide when I find it, but that's how I would answer this question. And this is the kind of thinking, you kind of have to sketch things out. Maybe you don't get all of this, maybe you get part of it, but I'm sure you could have gotten some of these answers. Let's go ahead. And it looks like you're going to get 1 point for each of these. One point for this and 1 point for that. All right, so this looks like it's that molecular genetics business. So here you have gibberellin, and it's a primary plant hormone. It promotes stem elongation, and beta-hydroxylase is an enzyme that catalyzes the reaction that converts a precursor gibberellin to the active form of gibberellin. The mutation GA3H gene results in the short plant phenotype. So let's go ahead and make sure that I'm kind of highlighting what I think is important. Again, as asked by one of your colleagues, you might want to go ahead and read the questions first and then come back and see what's important. But I like to, this is my preference, and I would definitely suggest reading the questions first. It does make sense. I'm just used to reading the questions and trying to figure out what's going on, and then going to the question and coming back to the reading. That's how I do it, but that's me. Gibberellin is a primary plant hormone that promotes stem elongation. GA is an enzyme that catalyzes the reaction, right? So there's a hormone that promotes elongation. There's GA3 beta hydroxylase that catalyzes that reaction and converts a precursor of gibberellin. So you have this, let's go ahead and write it out. You have I'm just going to call it pre-gib. You got some kind of reaction that makes gib. I'm just going to write gib because I'm not going to spell that whole big word. And the enzyme that catalyzes that reaction is GA3H, right? I put it above the arrow because it's not involved in the reaction. The mutation in GA3H results in a short plant phenotype. So this again, now gib, what's the result of that, by the way? And I guess I should, because this is an enzyme, gib is a hormone rather, and I can go ahead and label it hormone. When this hormone is present, that equates to tall plants, right? Why? Because they increases elongation, increases growth, height. Okay. When a pure breeding tall, converses the mutation GA3H gene results in a short plant. So there's some kind of mutant GA3H, and I'm just going to put use a different color, I suppose. You have mutated GA3H. Somehow, they don't tell you what it does to the hormone. There's a question that leads to this short, right? So it must be impacting elongation. There's an inhibition to elongation. It still happens because you got a short plant, so it must have grown to some extent, but it doesn't, not as well. All when the pure breeding tall plant is crossed with a pure breeding short plant, so there's a pure breeding, so that means in Ga3H, let's go ahead and say the wild type is, let's call it G, little g for, big G for the pure breeding tall plant, phenotype tall, is crossed with a pure breeding short plant. There. There's an assumption that the reason they're tall and short is because of the big G and little g. They don't actually state it, but that's fine. So when a pure is crossed with a pure breeding, all offspring in the F1 generation are tall. When the F1 is crossed with each other, 75% tall, 25% short. Now this is the classic example. This, I believe, what is on Bully 1. It was a genetics, molecular genetics problem, right? So this is just a classic example of dominant and recessive, right? So what's going to happen when you cross these two is you're going to get 100%. 100% are going to be big G, little g. And of course, those are all tall. But you take two of those, big G and little g, again, both being tall. You cross them, and you're going to get, and do the Punnett Square if you don't believe me. In fact, I'll just do the Punnett Square here on the side. Big G, little g, crossed with big G, little g. You see two big G's, a big G, little g, a big G, little g, and little g, little g. It give you 75% tall. And you'd have, the result would also be, this is the result, 25% short. So just, there would be 75%, or 3 out of 4, 3/4, 75% tall, and 1% short. So I'm thinking about all this. This is what comes in my mind quickly when I'm reading the problem. It says, the question goes on, it says, that's all the given, right? All this is what's been given to me. Now let's go ahead and read the question. The wild type encodes for GA3H enzyme with alanine. So the wild type allele, which is the one with that, that's not the mutant. So when they use the word wild type, they mean not the mutant, right? So the wild type allele it has alanine in it, a non-polar amino acid, so I know it's non-polar. And the position of that is at position 229. You have at position 229 you have alanine, a non-polar amino acid. A mutant allele has threonine. So now the mutant allele, the M, let me use brown because that's what I used for the mutant, the previous. So brown, threonine at position 229, and that's a polar amino acid. So the big difference between this in this amino acid chain is here. This is the big difference. Describe the effect of the mutation on the enzyme and provide the reasoning to support how this mutation results in a short plant phenotype in homogeneous recessive plant. So it goes back to the folding of proteins. So you have amino acids, right? and a polypeptide or protein and a hormone is in this case, this protein's a hormone. You have amino acid 1, amino acid 2, etc. And there's a ton of them. And then you have amino acid 229. Now, amino acid 229, and then of course it would continue, or maybe it's the end, but likely continues. So maybe amino acid 229 is a non-polar amino acid. It's this in the wild type, in the GA3H, it's alanine. It's non-polar. Where in mutant GA3H, or the non-wild type, right? It's not alanine. It's threonine. So it's THR. And this is polar. So the protein's folding, and as it's folding, perhaps you have the normal protein would look something like, let me go ahead and draw it in yellow. The normal protein would look something like this. The hormone would look something like this with alanine pointing inside, so it's wrapped around like so, because it's nonpolar. But now threonine comes along, you replace it with threonine, now it takes on a different shape. Now it folds differently. Maybe it folds like this. And this is threonine. It's polar, so it's attracted to water. Water's surrounding it. Remember that in all living systems, you have water out here. And so what this protein, what alanine is going to try to do, alanine is going to be repelled by water. And so because alanine is going to be repelled by water, it's going to sit inside a hydrophobic space, right? It's not going to be, the hydrophilic amino acids are all pointing out, where the hydrophobic ones are pointing in. So now threonine comes along. Threonine is polar, so it's hydrophilic, so it's pointing outside. So you've changed the shape. The point is, by changing one amino acid, you change the shape of the protein that's folding, and that causes the hormone not to work, or not to work as well. So by changing amino acid 229 from alanine to threonine, from nonpolar to polar, the shape of the hormone has changed. This has decreased its ability to affect growth or elongate. Negatively. You can say decreased. You have to make sure you're saying that it's decreasing it. They have to know that you understand that it's that this change is decreasing it. Yes, I understand that it's likely you're going to say, well, that's common sense. They don't care about common sense. What they're caring about is that there's sufficient evidence that you understand the problem, and that you're able to correctly identify the impact of changing a non-polar amino acid to a polar one. If you can do that, then they're fine with it. They'll give you the point. If you can't do that, they won't give you the point. I mean, it just turns out to be that simple. All right, so and I suppose if you have, and to be fair, you should, I would include if both alleles have this mutation, then the only form of the protein present in the plant is this less effective hormone. Remember, hormones are cell signals, and hormones, like all signals, are going to be based on this idea of shape matching shape. This question says, using the codon chart provided, predict the change in the codon sequence that resulted in the substitution of alanine for threonine at amino acid position 229. Well, you don't know what the code is, but let's go ahead and look at alanine. How do you get alanine? That's ALA. Let's see here. There's alanine, right? So any one of these will get you alanine. Are there any other alanines? Let me see. Let me double check. I suppose after all these years I should have memorized this, but no. And the substitution went from alanine to threonine, THR. So let's go ahead and find THR. Let's go ahead and use pink for THR. So there it is. THR forms that. Now I want to point something out here. In order to get THR, you need ACU, ACC, ACA, and ACG. That's why they chose for this question alanine and threonine, because the way you get alanine to change to threonine is simply by changing the first position from a G to an A. No matter what the other two are, the other two positions in the codon are, you can see it can be CU, CC, CA, CG. If the first position goes from G to A, you go from alanine to threonine. That's it. You have a mutation in the first position of the codon from G to A. It goes from alanine to threonine. And that's it. That's all you have to say. Now, what I just said is based on the idea of the codon, but of course, if you wanted to, you could discuss. What the heck is going on? Forgive me, I don't know what just went on. But if we go ahead and we look at this, and we say, I just said you just switch the first position. I described this in the terms of codon. Now, could you say then that if you had an RNA 5' to 3' that was labeled GCN and that you got there was a change in or a mutation that changed that amino acid, that codon to, I'm sorry, instead of GCN, ACN 3'. Could you give specifics? Sure, you could do that. I'm not sure if you need to go through all that trouble. I just think that if you just keep it simple and just use the evidence that you have in front of you and say that the first position of the codon changes, any of the first position of the codons change from G is the only way to get alanine to an A, then you would go from alanine to threonine. Honestly, I think that's the best answer. There's a couple ways you can look at it through DNA, through the you can describe RNA that changes in the RNA, mRNA changes in the DNA, but really just looking at the first position of the codon, describing that, I think is sufficient. So here, the last question here is just asking you to describe what I actually already described in the answer to A a little bit. If you looked at A, I said if both alleles have the mutation, then the only form of the protein present in the plant is this less effective hormone. So C is asking you to describe what would happen. Why is it that if you have a big G, little g, you get this tall, but if you get a big G, you also get tall. How is that possible? Well, the way you would explain it is to explain that if big G is present, let me just go ahead and use the terms that they would want you to use. If the wild type form then there will be sufficient levels of functional, I should say fully functional hormone for tall plants to form. Whether that be one copy or 2 copies of the wild type genes. The lower limit of functional protein has been reached. All right. Might want to talk about enough enzyme to produce, I'm not going to rewrite all that. I'll just say you might want to talk about enough enzyme is produced, functional enzyme is produced to produce enough hormone. Because remember, the mutation is actually not impacting the hormone, but it's impacting the production of the hormone, right? So this enzyme, the mutation's here, and so if you get a mutation here, you're not producing enough of the hormone. If you have even one wild type copy, you're producing enough hormone, functional hormone, or functional concentrations of the hormone to get the tall plant, even if you only have one enzyme. But I think it might be important to mention it is the enzyme, so they know that you're understanding. You understand that it's the hormone, that this enzyme is producing the hormone that's increasing the elongation of the plant so that it becomes taller. And so it's important, I think, that you mention this enzyme portion, this enzyme part of the discussion. Well, in this case, we're looking at a food web, and it's a really interesting way of looking at a food web. So I'm looking here, dietary composition of organisms in aquatic ecosystem. I know food web, and I thought food web, and I'm not sure, but I'm pretty sure it's a food web because I'm looking at trophic level here, and I see increasing trophic levels. So I know automatically that my producers are at the bottom. Maybe I should use green for producers. Producers at the bottom, and my consumers are my primary, secondary, tertiary, and then consummate or apex predator. Your top tier consumer is at the top here. That's what I'm thinking. But then this is a food web, it looks like. It looks like midgets are feeding everybody. And I know that the direction of the arrow is the direction of the energy. So these midgets are feeding whatever goes here. This midget's feeding whatever goes here. This midget's feeding whatever goes here. And whatever goes here is feeding the midget, because see, the arrowhead is pointing towards the midges. Not midges. All right. So I see algae, food source, percent of diet. Algae doesn't eat anything. They're producers. So algae is a producer. Stoneflies eat midges. Midges eat algae. They don't eat anything else. Stoneflies also eat caddisflies. Helgrammates eats stoneflies and midges and caddisflies. So that's probably closer to the top. And caddisflies eat algae and they eat midges. All right. So what's the only thing that midges eat? I'm going to have to say it's algae. So I'm going to go ahead and put algae down here. I don't have to worry about midges. Midges are there. So I don't have to worry about algae. I don't have to place algae. I mean, I shouldn't put a line through it because I don't know if I have to. If they're what part B is asking me to do. Stoneflies eat midges and caddisflies. So they're eating two. So they're higher than midges, but threes. And I know I'm done with midges. But Helgrammates is probably at the top, right? So Helgrammates is going to be at the top because it's eating three different consumers. It's not eating a producer at all. It's eating three different consumers. This is the producer, remember. So this one's probably up here at the top. Now, when I'm looking at caddisflies, they're eating 70 of them do eat algae, and then some of them eat midgets. So they're probably lower on the trophic level. So I would have to say that caddisflies that eat algae and midges are probably going to be there. And if it was going to be an accurate one, you'd obviously have to draw a line here, an arrow there, that kind of thing. And then the last one is a stonefly, and that would go here. I believe that would get you number 4. Oh, I filled that in without even reading the question. Jeez. So the table above shows blah, blah, blah. The rows represent organisms. I hope I'm right. I'm sorry if I'm wrong here. I just assumed. The rows, the columns represent food source. The percentages indicate the proportional dietary composition. Each organism, that's what I thought. Yeah, percent diet. So, I mean, that's just describing the chart. Based on the food source indicating the data table, construct a food web in the template below. Write the organism's name on the appropriate lines and draw arrows necessary to indicate the energy flow between organisms in the ecosystem. Oh, all right. So I do have to actually draw the arrows. All right. So caddisflies do eat midges, which is that arrow is already here, so I don't have to draw it. See, this is this arrow. Caddisflies eat midges, but they also eat algae. Caddisflies eat algae and midges, so I have to draw an arrow pointing upwards to caddisflies. So caddisflies are done. Grammatis or whatever it is, they eat midgets and they eat stoneflies and they eat caddisflies. So stoneflies feed that and caddisflies feed this as well. Now that takes care of that one. Midges eat only algae. Okay, so that's taken care of. Now, stoneflies eat midges and caddisflies. So they eat midges, which is, again, that's that arrow right here. But they also eat caddisflies. So notice the arrow's going to what is pointing. The arrow's pointing to what is eating the thing that where the arrow originates from. And, you know, I don't know, maybe I'm being a little picky, but I'm going to go ahead and draw this way. I don't know, I like that. It's a little neater. And then algae is feeding the caddisflies and the midges. I don't see, and that's all that algae's feeding. All right. I think that would get you your points for A. Probably get 1 point for putting the organisms in the right place, another point for putting the arrows in the right place. If you put the organisms in the wrong place, you're going to put the arrows in the wrong place, so you get 0 points. But you could put the organisms in the right place and then mess up the arrows and only get 1 point, right? So you can still pull 1 point just by getting them organized in the right place, even if you mess up the arrows. Which the biggest mistake people make with these arrows, by the way, it's a common mistake, is drawing the arrows in the wrong direction. That's really kind of a silly, simple thing. I mean, anybody can make that mistake. It's just something you might want to check before if you do have to draw a food web, that you get that right. In an effort to control the number of midges, an area within the ecosystem was sprayed with a fungus. With the fungus, there it is, which significantly decreases the midge population. So the fungus attacks the midges. So you're going to add, I'm going to go ahead and just draw it in. So this fungus is going to attack and kill the midges, right? I'm not even going to draw an arrow, because if I draw an arrow, I'm going to confuse myself. Maybe a double arrow, or I don't know. Let me draw a Pac-Man. Teeth. Gonna eat the midges. It's gonna go to town up the midge population, and based on a table, predict whether the spraying the fungus will have the greatest short-term impact on the predict whether the spraying of fungus will have the greatest short-term impact on the population of stoneflies, caddisflies, or helminthides. Justify your prediction. Well, which of these in the short term, so would this fungus impact of these that they listed? And they listed stoneflies, caddisflies, and helminthides, those three. So of these three, stoneflies, caddisflies, and helminthides, which is going to have the greatest short-term impact on them? Well, if you think about it, if this fungus gets rid of all the midges, the caddisflies still eat algae, so the caddisflies could still survive. To lesser extent, obviously, because they don't have midges. But if the midges aren't eating algae, there's probably more algae, so I don't know. Stoneflies, however, are eating two things. They're eating caddisflies and they're eating stonefly, and they're eating midges. And if you look at the numbers, look at stonefly, 90% of their diet is midges. Only 10% is caddisflies. And look at caddisflies, 70% of their diet is algae. Only 30% is midges. And helminthes, or however you pronounce Helgrammites, 70% of their diet is caddisflies, and another 20% is stoneflies. Only 10% are midges. So the one that's going to be impacted the most is going to be stoneflies. And so how would I justify? I would say stoneflies, because 90% of their diet is midges, where the other two, caddisflies and Helgrammites, have only 10 and 30%, or 30 and 10% respectively. Only have 10 and 30% of their diet from midges. So I think the answer is clear that it would be stoneflies. Okay, so here's that algae bloom and cyanobacteria and decomposers, right? An oxygen level. And this is that whole eutrophic lakes and the impact of fertilizer and other human products, putting sewage, for instance, raw sewage, into bodies of water, lakes and rivers, even oceans. When we dump a lot of nitrogen, whether it comes from poop or processed poop like fertilizer, or synthetic fertilizers that have nitrogen, potassium, and phosphorus, when these nutrients reach these bodies of water in unnatural concentrations, what happens is, or I shouldn't say unnatural, but mostly unnatural, but certainly in larger than normal concentrations, what happens is that the following, right? So you have relative concentration of oxygen, cyanobacteria, and decomposers. So cyanobacteria, algae, very similar to algae, they start to increase in number in stage 1 here. And as they increase in number, the oxygen level goes up. That makes sense because cyanobacteria are making oxygen. Yes, great. But at some point, the population becomes too large. It's no longer sustainable. It's reached the carrying capacity of the environment, however you want to look at it, and they start to die. So as these cyanobacteria start to die, as their concentration come down, the decomposers go up, because the decomposers are eating the dead bodies of the cyanobacteria or algae or whatever it is. And the decomposers go up. Then the decomposers are using the oxygen because they're doing cellular respiration. The algae are doing photosynthesis. That's why oxygen is going up. But the decomposers are using oxygen, producing carbon dioxide. So the oxygen level is going to go down. It's going to crash. Now that's what's happening at the levels of single-celled organisms, right? So because of this population dynamics between these predator-prey dynamics that you see here between the decomposers, and it's not really predator-prey because the decomposers don't prey on cyanobacteria, they just decompose cyanobacteria. But because of the population dynamics of the cyanobacteria and then the resultant decomposers, you're getting a change in the oxygen concentration in the water, which of course is going to decrease the concentration of the water, decrease the available oxygen by the fish, the available oxygen for the fish in the water, so the fish die. I mean, that's what you would expect at the lower levels of oxygen once you go past what you would have before these events happen, right? So probably at this point is where you'd probably start to see some fish dying. And if it gets to zero, well, that's it. Dead lake or river or whatever. Probably a lake is going to have the biggest impact there, because moving water is going to have some oxygen in, just by the mixing with air. So Microcitus aeruginesis is, and I know I'm mispronouncing it. I never know how to pronounce these words. Is a freshwater photosynthetic cyanobacterium. The temperatures increase and nutrients are readily available in its pond habitat. So when temperatures increase and nutrients are readily available in the habitat, this undergoes rapid cell division and forms an extremely large visible mass that's called an algal bloom. So when these get together, there's so many cells that you start to see them floating on the water, and I've shown you pictures of this in class. So as M. aeruginesis has a short lifespan and is decomposed by anaerobic bacteria, anaerobic meaning it uses oxygen, right? And fungi. So the fungi and the bacteria that use oxygen eat the algae when it dies. Identify the metabolic pathway of the organism that is primarily responsible for the change in oxygen level in the pond between 1 and 2. So this 1 is going up. In between 1 and 2, oxygen is going up, right? At 2, it starts to go down. And so between 1 and 2, it's photosynthesis. Between 2 and 3, it's cellular respiration. Photosynthesis by cyanobacteria and cellular respiration by the fungi and aerobic bacteria. Well, that's an easy answer. Let's go to the next one. So here you have a cell with DNA damage, and then there's individual nucleus placed on a slide. So they took the nucleus out of the cell with DNA damage, maybe too much sun, maybe some kind of chemical, maybe a virus infected the cell's nucleus, whatever the case, or bacteria, who knows. So the nucleus is pulled out. The bacteria probably wouldn't get into the nucleus; it'd be here in the cytoplasm, but whatever. So the nucleus gets. For the record, I'm sitting here in office hours and no one's joined yet. I'm in 5th period now. No one's joined for office hours, so no one seems to have any questions. Well, I hope that's good news. So in any case, you have an individual nucleus is placed on a slide. Then you do electrophoresis. There's a head and a tail. Intact DNA remains in the head, and the damaged DNA is in the tail. Right. So this is electrophoresis. The head and the tail on. Remember, DNA has negative charge, right? So DNA has an overall, and I haven't gotten to this part I haven't read the problem yet, but you have DNA, and DNA has an overall negative charge on it. So the movement of this molecule is going to be in this direction through the gel. This is where you get damaged DNA is probably going to be uncoiled, right? So if you have the intact DNA is going to be here in the head, and then this is according to the reading, and then the damaged DNA is here in the tail. That's what I'm reading so far. I need to know that DNA has generally a negative charge, and I should know that because it has a sugar phosphate backbone, and phosphate is oxygen, 4 oxygens on a phosphorus, which is negative, and since it has a sugar phosphate backbone, you have a generally negative charge to a DNA molecule. All right. So The nucleus of an individual cell is placed on a microscope slide coated with an agarose gel. The electric current is applied to the gel and causes DNA to move, electrophoresis. The DNA is stained with a fluorescent dye. When viewed using a microscope, undamaged DNA from the nucleus appears to be round shape. The head and the fragments of DNA extend out from the head. The tail, the length of the tail corresponds to the amount of damage in the DNA. Okay. Explain the movement of DNA fragments in the comet assay. Identify one property of DNA to provide the reasoning support how the property contributes to the movement during the comet assay technique. I already answered it, oddly, that DNA has a negative charge, so it's going to move towards the positive end. And of course, specifically a comet, and they call it a comet, by the way, because remember, a comet, coming to Earth has that tail of gases as it goes around the sun. In its orbit, a tail is formed in a comet. So that's why, and that's what when you see there's a head and a tail, just like in the comet, there's a head and a tail. The tail is moving faster. And it's theoretically it's because they're fragments, right? DNA fragments that have broken off. This DNA damage is the breaking of DNA, and so the little broken pieces are moving faster through the matrix. And this goes back to how gel electrophoresis works. Let me quickly remind you how gel electrophoresis works. You have these gels. These agarose gels are a matrix of seaweed proteins, right? Agarose is seaweed proteins. Gelatin that you eat, or some people eat, is made from pig marrow, but the protein, the process is the same. It forms this gelatinous mass, which is these proteins that bind across their lengths, and they form these tunnels. And the tunnels are random in size and shape and direction. And so when this protein, you have two proteins, this protein versus this, let me choose another color, versus this protein. This fragment, if this is a big ball of DNA, right? This is a big ball of DNA. It's going to go through this maze slower. It's going to go through this maze a lot slower, as it has to choose spaces that are fairly large, where the smaller DNA can move much quicker through the pores in a much more direct route. So when you're looking at it from the microscope scale, and this is actually at the very, very tiniest scale, at the molecular level, right? What you would see at the microscopic scale is you'd see the big ball of DNA up here, and then as the fragments got smaller, they move further down the gel across towards the positive end. As time went on in this electrical field, they would move towards the positive end, so the smaller fragments move along the tail. So those are the two things, is the negative charge of the DNA, and the second point, and I think that would give you so because they're asking for one property to explain the movement, that's it, you're done. But in case you wanted to say the size of the fragments, are smaller than the undamaged, therefore unbroken DNA molecule. And therefore, and so move faster, move further in less time. Faster, right? So you get this comet tail look to it because you get this comet tail look to it because the small pieces move further. All right, so in a different experiment, cells are treated with chemical mutagen that causes only one nucleotide substitution in DNA. Predict the likely result of the comet assay for this treatment. So in a mutagen, now this is DNA damage, right? Here you're talking about DNA damage where you're breaking the double-stranded DNA, right? So you have this double-stranded DNA molecule, and there's in humans there'd be 46 or 23 pairs of these, and what we're talking about with the damage of DNA is you're talking about a break, right? So now you have these random breaks that break the double-stranded bonds and so you got two pieces left, right? So in the first example, in A, when we're dealing with A, you're talking about getting a piece of DNA that's this big, and it'll be more than a piece, it'd be several pieces, it'd be a ball, basically, and then you have all these little fragments that broke off, and the smaller the breaks, the further they go, and so you're going to get this big ball of DNA that's not damaged, and then you get these little pieces of damage. They're going to go further in less time. Okay, so that's A. But in B, what they're saying is that what happens is instead of breaking it, what if we just substitute it? What if the damage is not breaking but substituting? Well, there'd be no change in the size, right? You're not breaking it, so all the molecules would be the size that they would normally be. So you would get one ball of DNA. It wouldn't be in this particular type of assay where you're not breaking them up. You're not separating them by size because they're all the same size. So you'd end up with either one big ball, right, of DNA, the head, no tail, right? So you'd end up with a head or no tail. Yikes. You end up with a head with no tail. Or you'd end up with maybe a shorter tail, because maybe you do get a few breaks. I'm not sure, but I would go with head or no tail. That's probably your best answer. I guess you could think of it as having a shorter tail, depending on. Certainly, your tail would be shorter than you would expect if you're introducing double-stranded breaks. The best answer in the particular way they phrase the question, I think the safest answer is that there would be a head with no tail. It's a lot of questions. So many species of bacteria grow in the mouths of animals and can form biofilms on teeth or plaques. Well, we all know that. We go to the dentist. Within plaque, outer layers contain high levels of oxygen, and the layers closer to low levels of oxygen. The surface of the tooth is covered by a hard layer of enamel, which can be dissolved under acidic conditions. When enamel breaks down, the bacteria in plaque can extract nutrients from the tooth and cause cavities. All right, so again, with me, I like to draw it out, and I mean, if it works for you to do it another way, I'm not gonna argue it, but I would have to see the evidence of it working for you. So the teeth have these roots. I'm sure you've seen something in different shapes. It's horrible tooth, but whatever. Let's just go ahead and draw kind of a molar rather than some kind of canine. So you have this white enamel, right? That's what I imagine when I think of a tooth, and then I see. So this white tooth. Let me go ahead and spend the time drawing the tooth out. how much time you have is obviously going to define how much time you have spent, drawing it all out. I wouldn't spend much time at all drawing it out like this, but just for the sake of our discussion, I'm going to go ahead and draw it out. And then I would expect you to have some kind of pink gum line, right? So there's some gum line here. So your tooth sticks out. The outside's enamel. The roots feed the living part of your tooth. Your tooth has a living core, but the outside is a hard enamel. The tissues, the gum line is living tissue that helps support it with bringing blood to the living part of your cell, of your tooth. All right. And then they say there's this plaque, right? And they say that these layers of plaque exist, and they turn your teeth yellow. So there's this layer of plaque. It kind of. I wonder if I can change the opacity. Let me see. So in changing the opacity, I think we can kind of think of this layer of yellowness around the tooth, these plaques that develop, these layer of bacteria that grow. And there's layers of it. They're saying that you have kind of an outer layer. If you did a cross section of this, you'd have a little hard layer, then there'd be another layer of bacteria, another layer of bacteria, another layer of bacteria. Then you'd have your tooth surface, right, the enamel surface. All right. Identify. All right, so certain types of bacteria, Streptococcus mutans, thrive on innermost anaerobic layers. So I know that this is going to be anaerobic, so you're not going to produce. They're not going to use oxygen. These bacteria out here. I'm going to use a different color. These bacteria out here use oxygen, and these bacteria down here. Let me use no oxygen, right? No oxygen is necessary. So these are anaerobic, and the ones on the surface are aerobic, right? So there you go, a spacer. All right, so of the plaque, and they're associated with cavities. All right, so they're this anaerobic pathway, so they're associated with drilling or burning or corroding holes in your teeth. So one would think that kind of activity it's because these anaerobic bacteria producing no oxygen, but they are producing acid, right? Some kind of acid. And you can read that they say this is, acidic conditions can be dissolved under acidic conditions, and they tell you that the enamel layer can be dissolved in the anaerobic bacteria of the plaque associated with the cavities. So that means that, the anaerobic bacteria, and we can use a different color maybe. These are the bacteria that live down here. These guys are anaerobic, and they can produce acids that eat away your enamel. All right. Other types of bacteria, Streptococcus sanguinis, compete with S mutants, but are unable to thrive in acidic environments. So even though there's this other Streptococcus, and both Streptococci are little circular bacteria, but they're different bacteria, right? So they're competing with each other. This Streptococcus sanguinis lives here. Out here probably outcompetes, but the Streptococcus mutans beats out the Streptococcus sanguinis because there's acidic conditions. It's producing some kind of acid down here, where the mutants can survive in those acidic conditions. All right. All right, so identify the biochemical pathway for S mutants and uses for metabolizing sugar, and describe how the pathway contributes to low pH in the inner layers of plaque. It's fancy. Sounds fancy indeed, but it's not all that fancy. So anaerobic pathways. So we're talking about what's an anaerobic pathway we've learned about? Fermentation. So let me review for you. So when you're dealing with fermentation, you're dealing with sugar comes in. So sugar comes in. All sugar goes through glycolysis. Produces NADH, FADH2. Your electron carriers and ATP. And of course, the final product of glycolysis is 2 pyruvates. Now, pyruvate has 2 options. In the presence, if oxygen is present, then to the mitochondria. This is in eukaryotic organisms, right? Obviously, these bacteria don't have mitochondria. And then the other pathway, that's one pathway, where they can do, in the mitochondria, can do aerobic respiration, is if there's no oxygen present, then it's going to do anaerobic respiration, what we call, if no O2, then you're going to do what's called fermentation. Either you're going to produce alcohol or an acid called lactic acid. So that layer of plaque at the bottom of all these layers of plaques, all these sheets of bacteria, they're doing anaerobic respiration, and because they're able to produce all these acids, they're beating out all the other bacteria, and they're multiplying. And so they're doing fermentation, and they're producing lactic acid. So identify biochemical pathway for metabolizing sugar. Describe the pathway contributes to low pH in layers of plaque. Fermentation, lactic acid is produced. And you're done. I mean, I think that would answer the question. All right. Normal tooth brushing effectively removes much of the plaque from the flat surfaces of teeth, but cannot reach the surfaces between the teeth. Many commercial toothpaste contain alkaline components which raise the pH of the mouth. Predict how the population sizes of mutants in S. sanguinis in the bacterial community in the plaque between the teeth are likely to change when toothpaste are used. So obviously when you're dealing with the spaces in between the teeth, and this is again where the drawings come in handy, when you're dealing with a tooth, the 2 teeth sitting next to each other, two teeth sitting next to each other along the gum line. Let me do pink because I'm sorry, I'm going to get a little anal here. I just want to be consistent. All right, so brushing gets rid of the plaques on the outside here, so you're not producing. So there's oxygen present, so there's no lactic acid production, so you don't get less cavities. That's how brushing helps. But then it can't reach in between. But then if you add, toothpaste has this basic component, which you know is going to neutralize the acid, by the way, if it can reach it. But remember what they said up here about sanguinis and versus the mutants, the mutans. The mutans thrives in acid, but the sanguinis is downregulated by acid. It can't compete with sanguinis in that acidic environment. So now you're getting rid of the acidic environment, so now you would expect sanguinis to increase in population, and you'd expect mutans to decrease in its population. So you'd expect sanguinis to go up, the population of sanguinis to go up, and the population of mutans to go down. Easy question. I guess I gave you the 3 hard ones, and I might as well give you some easy ones here. As mutans decreases, notice I labeled them, and S sanguinis, trying to see how to spell it, G-U, increases in the less acidic environment. That's it. Those are your 2 answers. I think that would satisfy me, and I think that would satisfy them. You have to look at the, it says predict. Would, maybe use the word would, decrease. Just so nobody gets upset that you use the wrong verbiage. Would. I can't see them actually saying anything about it, but whatever. Just make sure it says predict. So say would increase, would decrease. All right, so I think that would do it. Let's look at the last question here. Oh, it is the last question, thank God. It said estrogens. I can't read anymore. Estrogens are small hydrophobic lipid hormones that promote cell division in the development of the reproductive structures in mammals. Estrogens passively diffuse across the plasma membrane and bind to the receptor proteins in the cytoplasm of target cells. All right. Again, I'm just going to go ahead and draw it. Nucleus, blah, 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 blah. But there's going to be a receptor here. And the estrogen, estrogen is going to come in. Let's just use a ball. Estrogen's out here. It's going to passively diffuse, so high to low. And it's going to go and meet up with the receptor. And it's going to, what's it doing? It's going to increase cell division, and it's going to help develop reproductive structures in mammals. So estrogen is going to help produce testicles and ovaries. All right. Describe one characteristic of the plasma membrane that allows estrogens to passively cross the membrane. My goodness, if you get this one wrong, I don't know. I think it's probably the easiest answer they could. I would hope they give you a question like this. Well, the plasma membrane is hydrophobic. Like dissolves like. Right? So the plasma membrane is hydrophobic. This allows small hydrophobic molecules to pass through. There's one. That would give you 1 point. There's no doubt in my mind. But there's another, or you might want to say that the space between phospholipids. I honestly, this is the answer, man. And guys, this is the answer. This is the answer you want to remember. It's like dissolves like. It's going to be the answer more often than the space between the phospholipids allows. That this one is actually probably more true for things like water that's hydrophilic, but it's small enough that it can kind of leak through the membrane in between the spaces. Not very well, but then you have aquaporins that let the water in as well. So you do have some channels for water in your cells. But when you're talking about space between the phospholipids, it is a way for things to get into the cell. And I can't not mention it, but the plasma membrane is hydrophobic and allows small hydrophilic molecules to pass through. That's the big one, right? All right. In the laboratory experiment, a researcher generates antibodies that bind to purified estrogen receptors. So you have some antibodies that bind to this. They're going to bind to that. Extracted from cells, the researcher uses antibodies in an attempt to treat estrogen-dependent cancers, but finds that the treatment is ineffective. Explain the ineffectiveness of antibodies at treating estrogen-dependent cancers. So it's going to increase. So estrogen is going to increase the cell cycle, right? The reproduction, the cell division. So if you have antibodies for this receptor, it would stop the receptor. If you could bind the antibodies to the receptor, it would bind to the receptor and it would stop estrogen from working. That works. That would stop it. But what's the problem here? Well, again, my drawings are going to come in handy. So, and this is the difference, by the way, between in vitro and in vivo, right? Vitro in a test tube, in a lab, and in vivo in a living organism, a living system. Now, in a test tube where you take and you have the estrogen in receptors isolated, and there's all these, and then you add antibodies. These antibodies come and bind to them. For sure, you can add estrogen all day to these and they're not going to bind because they're surrounded and bonded and the antibodies have bonded to the receptors. So you can add estrogen all day long and there's no way they're gonna match up with their, I should do pink. There's no way they're going to be able to bind to the receptor because the antibodies would block it. Well, that would work in a test tube. But in a living system, you would have antibodies are produced by cells and they travel in the blood. And the antibodies that you would inject someone with, or you would allow your blood to be filled with these antibodies, are great if the receptor were in the blood, but they're not in the blood, they're in the cells. So the receptors that the antibodies are made to are here in the cells. And they line the tissue, they line the blood cells, the blood vessels, they're in the tissues. How are the antibodies, or these large proteins, going to get to the receptor in the cell? How is that going to happen? Well, it's not. So, creating antibodies to intracellular structures don't work it doesn't work. Don't work. I'm so tired. I don't know if they can. Won't work because the antibodies are large proteins and cannot enter the cell. That's it. They can't get into the cell. So you can make all the antibodies you want, unlike COVID, which is in the bloodstream, and you can actually, the antibodies would work against a virus, because as soon as the virus comes out of the cell, the antibodies would bind, and the immune cells of the body would destroy them. Here, the receptors inside the living cell, and these antibodies would have to pass through the membranes, and that would just be way too difficult, way too slow, and it just wouldn't work. These cells would continue to do their uncontrolled mitosis because estrogen would be in the blood and then would pass through the cell with no problem. So estrogen that would be out here, the hormone would be out here, and that would still get in and still stimulate cellular division, and so you would get cancer to occur. Now, one possible solution is to make antibodies to the estrogen, but that won't work because you need estrogen for normal function. So you can't really impact the cell. You can't really impact cancers by making antibodies to estrogen or the receptor. You have to find another method. So for this type of cancer that's dependent on estrogen. All right. So that solves that problem, and again, that's mostly just drawing and thinking about it. That one's a little bit harder, a little bit more thinking involved. Maybe you only got one, A and not B or whatever, but hopefully you certainly got A. And B wasn't all that hard. You'd have to know that an antibody is a big protein is the only limitation on that question for you to be able to answer correctly. All right, well, have a good day. Hope this helps. And I'll find the answer key as well and post it as well. All right, bye.